Comprehensive chromosomal screening is the process of evaluating an embryo to determine if it has the right number of chromosomes in it. As it turns out, humans are not very efficient at going from a full set to a half set of chromosomes. Now, why is that important? In a young lady, her oocytes begin with 46 chromosomes, 2 of which are X. Little girls are 46XX, and we all had that in high school biology. The reality is that's perfect for every cell in her body except an egg. It must remove or throw out half a set of chromosomes so that the other half remains behind and can be eventually joined by a sperm, bringing a half set to go back to an entire new person. And so in that process of going from a full set, 46XX, to a half set, 23X, many problems arise. And in fact, when you're very young, errors can occur at 20 to 30% of the time. By your mid-30s, it's 40 or 50%. By the time you're your early 40s, it's 80 to 90% of embryos are chromosomally unbalanced. Now, these embryos look just the same under the microscope, and they develop just the same in the laboratory. And so it's important to evaluate the embryo directly, to allow the embryo to grow, and then perform a test called Comprehensive Chromosomal Screening on a few cells biopsied at the very end stage of in vitro development, called the blastocyst stage. When you do that, you contain very accurate information on the genetic makeup of that embryo. That's called Comprehensive Chromosomal Screening, and the advantages to doing it have been just overwhelmingly clear. Pregnancy rates go up, miscarriage rates go down. We take an average 42-year-old and make her Down syndrome risk less than it was when she was 21. All of that together allows comprehensive chromosomal screening to improve clinical outcomes for patients. They spend less time in care, they spend less total money on care, get healthier babies and better outcomes and fewer multiples. This is an extraordinarily important advancement in our field.